Hi, welcome to the next in our OSTR series on cutting-edge methodologies. Today, we're going to talk about proximity-dependent biotinylation for the characterization of protein complexes and the mapping of organelles. A standard experiment for characterization of protein-protein complexes involves pull-down of a bait coupled with mass spectrometry to identify the prey that co-purifies with it. This is a mass spec variant of a standard IP or pull-down experiment. These are nice experiments. They're relatively straightforward, routinely used. If one can do an IP Western, adding it into a mass spec detection isn't very difficult. And they're also readily coupled to quantitative methods to get relative information on changes in enrichment. However, there are some disadvantages. For example, you can lose interactions where an antibody binds if you're doing an immunoprecipitation. The antibody and protein interactors are going to compete for binding to the same sites, and the antibody is generally going to win. These experiments don't work well for hard-to-purify interactors, those that are transient or form unstable complexes. Also for enzymes, because the substrates of these enzymes are often low-affinity interactions. They're meant to be released and not stable. And also for baits that have poor solubility, such as proteins from membranes. Standard IP mass spec experiments are also subject to post-lysis artifacts. These are interactions that form only in the lysate. They don't normally form under physiological conditions between two proteins that aren't normally close together in the cell. So there are new ways that have been developed to try and overcome some of these disadvantages and help to find better protein complex information. The class of experiment that we're going to talk about today are proximity-dependent biotinylation, or PDB experiments. In these experiments, there's expression of a fusion protein of the bait with an enzyme that catalyzes activation of biotin or phenolic biotin derivatives. These are either going to be biotinligases or peroxidases. Expression of the bait fusion with the relevant context and using a biotin substrate leads to biotinylation of proteins that are close by in the cell. Lysis of the cell can then happen under potentially harsh conditions that you can get full solubilization of membrane proteins and capture of biotinylated proteins by streptavidin pull-down. This means that unstable or transient complexes could still be tagged and are still able to be pulled down. And so you can find interactors regardless of their affinity or their presence in the membrane. So let's look at some of these examples. Biotinligases are commonly used, such as the BRA or BioID tags. In these methods, there's activation of biotin to biotinoyl AMP. These enzymes have been modified so they're unable to catalyze direct transfer to a specific residue, but instead they release a cloud of activated biotin, which reacts with epsilolamines on lysines on nearby proteins, with an approximately 10 nanometer labeling radius. On the other side are peroxidases, such as the APEX and APEX2 enzymes, which catalyze tyrosine attack by phenolic compounds, which are coupled to biotin, which are activated by peroxide. These have a slightly larger labeling radius of about 20 nanometers. These are similar though, so the major difference in the two methods is the time required for sufficient signal for mass spec analysis. BioID experiments are generally performed with labeling of 12 to 24 hours, although the new TurboID and mini turbo tags can generate strong signals, reportedly in minutes. In contrast, the APEX2 methods, which use peroxide, are definitely performed on a time scale of minutes. So really the approach to go with depends on the type of experiment and what type of labeling speed you need. Just to go again into an example, as you can see on this slide, what you have is either BioID or APEX, where there are proteins in complex. When they're activated, those proximal proteins get biotinylated, and that can then be pulled down on streptavidin stepharos for mass spectrometry analysis. Since the streptavidin biotin interaction is so tight, these experiments can be performed under harsh denaturing conditions for the cell lysis and also under stringent washing conditions to help bring down the background. Looking here at the cell, you can see again, this is the scale on which things happen. The nucleolus is on the scale of 2 to 3 microns, so that when we're getting labeling radii of 10 to 20 nanometers, this really is a small labeling radius, and so we can get good modification within protein complexes or within organelles. There are some, of course, important controls and things to consider with these type of experiments, just as with any experiment. First is the background. There are, of course, natively biotinylated proteins in the cell, so one has to consider the ability to identify those as well. There are also going to be proteins that are promiscuously biotinylated due to some mislocalization of the fusion protein, as well as proteins that will non-specifically interact with the resin. So because of these proteins that contribute to the background, controls are very important. Controls to determine endogenous biotinylation, such as the bait protein, without addition of the enzyme or untransfected cells are often useful. Controls of the conditions that look at promiscuous biotinylation are also helpful. In this case, that would be just the enzyme alone, or it fused to an irrelevant protein that's unrelated to what you're studying. And furthermore, replicate analyses really help. These background proteins are going to tend to be low or random present, so that when doing replicate experiments, true interactors should come out time and time again. There are also some caveats to these experiments that are important to keep in mind. The residence time of proteins in the complex and the number of solvent-exposed reactive residues will somewhat determine how detectable proteins can be. Labeling also depends on the protein and complex side. So for example, if you have a very large complex, not all members will be labeled by a single bait, and so multiple different baits may be required. Further, for bait proteins that are embedded in membranes or localized to organelles, abundant proteins that are present in those locations but are not necessarily direct interactors will likely still be present and may dominate that identification list. Just because there's a lot of them, they're going to tend to be able to be labeled. So in that case, an extra experimental control in which proteins with the same localization are used but different interactors, or even a mutant form of the bait protein, may help to distinguish those possibilities. So this is an example using BioID, where the authors were using this method to look at proteins associating with mRNA biology, looking at specifically proteins in RNA granules, either stress granules or processing bodies. Using the methodology, they were able to find over 7,000 unique interactions with nearly 1,800 proteins. Analysis of the correlated patterns between the praise uncovered spatial organization of RNA regulatory structures and enabled definition of core components of both the stress granules and the processing bodies. So the method here was really applied nicely to these different subcellular localization groups and able to identify what actually constitutes the different bodies. In contrast, this is an example using APEX to study G protein coupled receptor signaling. First, the authors established that the APEX method could be used to characterize complexes of their GPCR after treatment with the agonist and with the antagonist, and identified approximately 1,200 protein interactors in the agonist-treated cells, including small g proteins. None of those interactors were enriched in the antagonist-treated cells. So knowing that the method worked, they could then use it to do a time-resolved experiment where they were able to track the interactions at 10 different time points, using 10-second intervals for the first 50 seconds and then longer intervals up to 30 minutes. In addition, they had one sample with no ligand treatment at all as a control. And so using the APEX method and the fact that it works over very short time scales, they were able to track proximity kinetics for over 1,000 proteins. They were able to look at endocytosis and subcellular localization over longer time scales, looking at the changes in the interactors known to be involved in the process or in the localization. And so this is a very unique experiment to be able to look at the effects of protein signaling complexes over time, look at how the complexes form and fall apart. And it's really a method that can only be done with this type of approach and experimental design. So for more information about these types of methods and to think about how you could use them in your own research, please feel free to contact your local mass spectrometrist. Within the NCI, you can talk to me, Lisa Jenkins, or my colleague, Thorkel Anderson in Frederick. Within the wider NIH community, I would direct you to visit the CREX for more information about available resources. Thank you very much for your attention.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4CANCER. Produced August 2019.